which scientists made significant discoveries toward the theory of photosynthesis.
The ancient Greeks and Romans believed that plants derived their food from the soil. The earliest experiment to test this hypothesis was performed by the Belgian scientist Jan-Baptiste van Helmont. 1577 to 1644, who grew a willow tree in a container of soil and fed it only water. At the end of 5 years, the weight of the willow tree had increased by 164 pounds. 74.4 kilograms, while the weight of the soil had decreased by 2 ounces, 57 gr. Van Helmont concluded the plant had received all its nourishment from the water and none from the soil. Joseph Priestley, 1733 to 1804, demonstrated that air was restored by plants. In 1771 Priestley conducted an experiment in which he placed a lighted candle in a glass container and allowed it to burn until extinguished by lack of oxygen. He then put a plant into the same chamber and allowed it to grow for a month. Repeating the candle experiment a month later, he found that the candle would now burn. Priestley's experiments showed that plants release oxygen, O2, and take in carbon dioxide, CO2, produced by combustion. The Dutch physician Jan Ingenhousz, 1730 to 1799, confirmed Priestley's ideas, emphasizing that air is restored only by green plants in the presence of sunlight. Evidence of photosynthesis's two-stage process was first presented by F. Blackman, 1866 to 1947, in 1905. Blackman had identified that both a light-dependent stage and a light-independent stage occur during photosynthesis. In 1930 C. B. Van Niel, 1923 to 1977, became the first person to propose that water, rather than carbon dioxide, was the source of the oxygen that resulted from photosynthesis. In 1937 Robert Hill, 1899 to 1991, discovered that chloroplasts are capable of producing oxygen in the absence of carbon dioxide only when the chloroplasts are illuminated and provided with an artificial electron acceptor.
What is an organ system?
An organ system is a group of organs working together to perform a vital body function. Vertebrate animals have 12 major organ systems.
Who was Nico Tinbergen?
Nico Tinbergen, 1907 to 1988, along with Konrad Lorenz, 1903 to 1989, and Karl von Frisch, 1886 to 1982. Laid the four cornerstones of ethological study: causation, development, evolution, and function of behavior. Tinbergen's earliest studies were of the aggressive threat displays of adult male three-spined stickleback fish, Gasterosteus aculeatus. Tinbergen found that by presenting the male with stickleback models, he was able to elicit an aggressive response. He began his career in animal behavior as a child by studying the mating and nesting behavior of sticklebacks in his backyard pond. He attended the University of Leiden, where he did his dissertation research on digger wasps. His thesis was only 36 pages long and was barely accepted by the faculty. In 1936 he met Lorentz at a symposium, and they became lifelong friends and colleagues. During World War II, Tinbergen was imprisoned in a German hostage camp and Lorentz in a Russian camp. When they were reunited after the war, both resumed their studies on animal behavior.
Do humans use pheromones?
Yes, human eggs release a chemical signal that allows them to communicate with sperm. Human females also respond to pheromones. In this case, the signals actually coordinate the menstrual cycle. In fact, 2 human pheromones have been identified. The first increases the likelihood of ovulation, while the second suppresses ovulation. For years, scientists debated the importance of pheromones to humans. But the close examination of the olfactory receptors and the vomeronasal organ in the nose has given researchers a mechanism by which humans may be able to recognize these compounds.
How does the immune system work?
The immune system has 2 main components: white blood cells and antibodies circulating in the blood. The antigen-antibody reaction forms the basis for this immunity. When an antigen, antibody generator, a harmful bacterium, virus, fungus, parasite, or other foreign substance invades the body, a specific antibody is generated to attack the antigen. The antibody is produced by B lymphocytes, B cells, in the spleen or lymph nodes. An antibody may either destroy the antigen directly or it may label it so that a white blood cell, called a macrophage, or scavenger cell, can engulf the foreign intruder. After a human has been exposed to an antigen, a later exposure to the same antigen will produce a faster immune system reaction. The necessary antibodies will be produced more rapidly and in larger amounts. Artificial immunization uses this antigen-antibody reaction to protect the human body from certain diseases. By exposing the body to a safe dose of antigen to produce effective antibodies as well as a readiness for any future attacks of the harmful antigen.
Can cobras really hypnotize their prey?
While hypnotism would be a fortunate side effect, it is thought that the semi-erect posture and swaying motion are probably used by the cobra to estimate the distance to its prey. African cobras don't strike at their target, rather, they spit venom at threats up to 6 feet 1.9 meters away. To do this, they need to take visual aim.
Who invented the thermometer?
The Greeks of Alexandria knew that air expanded as it was heated. Hero of Alexandria, 1st century CE, and Philo of Byzantium. Fluid ca 250 BCE, made simple thermoscopes, but they were not real thermometers. In 1592 Galileo, 1564–1642, made a kind of thermometer that also functioned as a barometer. And in 1612 his friend Sant'Orio 1561–1636, adapted the air thermometer, a device in which a colored liquid was driven down by the expansion of air, to measure the body's temperature change during illness and recovery. Still, it was not until 1713 that Daniel Fahrenheit 16861736 began developing a thermometer with a fixed scale. He worked out his scale from two fixed points: the melting point of ice and the heat of the healthy human body. He realized that the melting point of ice was a constant temperature, whereas the freezing point of water varied. Fahrenheit put his thermometer into a mixture of ice, water, and salt, which he marked off at 0 degrees. And, using this as a starting point, marked off melting ice at 32 degrees and blood heat at 96 degrees. In 1835 it was discovered that normal blood measured 98.6 degrees Fahrenheit. Sometimes Fahrenheit used spirit of wine as the liquid in the thermometer tube, but more often he used specially purified mercury. Later, the boiling point of water, 212 degrees Fahrenheit, became the upper fixed point of the thermometer.
How do plants maximize exposure to light?
The leaves of many plants move. Leaves often orient themselves perpendicularly to sunlight, thereby increasing the amount of light absorbed for photosynthesis. Leaves also form unusual patterns of layering, called mosaics, that minimize the shading of leaves by each other.
What is the difference between a territory and a home range?
A territory is a defended area. It can be as small as the space around a female red-winged blackbird's nest or as large as the backyard that your dog defends. A home range, in contrast, is simply the area where an animal spends its time. Home ranges may be shared with conspecifics, members of the same species, and may overlap those of other species.
What general characteristics do all fishes have in common?
All fishes have the following characteristics: 1. Gills that extract oxygen from water. 2. An internal skeleton with a skin that surrounds the dorsal nerve cord. 3. Single loop blood circulation in which the blood is pumped from the heart to the gills and then to the rest of the body before returning to the heart. 4. Nutritional deficiencies, particularly some amino acids that must be consumed and cannot be synthesized.
What groups of organisms have a cell wall?
A cell wall is present in organisms in the kingdoms Archaebacteria, Eubacteria, Protista, fungi, and Plantae. Animals are the only organisms that do not have a cell wall.
What is meiosis?
Meiosis is often referred to as reduction division, meaning that the number of chromosomes present is reduced from 2n, diploid, to n, haploid. The meiotic process consists of two separate cell divisions and occurs in the gonads, ovaries and testis. It is most important to sexual reproduction because of genetic variation that occurs as a result of this process.
How do cells become tissues?
Tissues are comprised of different types of cells that have a common purpose or function. Multicellular organisms are so complex that tissues are needed to perform vital functions such as support, transportation, and the movement of animal cells. In animals, the groups of cells that form tissue may become structurally linked via their cytoskeletons and membranes. As an example, epithelial cells are anchored to underlying connective tissue by a basement membrane. These cells are often exposed to mechanical stress, and a system of cell junctions between epithelial cells helps them respond when stretched.
What does this mean?
This means that structurally, the two strands of DNA, the two sides of the ladder, run in opposite directions. This property of antiparallelity allows for formation of the hydrogen bonds between the nitrogenous bases. These bonds are critical to the integrity of the molecule.
How does DNA correct its own errors?
Spontaneous damage to DNA occurs at a rate of 1 event per billion nucleotide pairs per minute. Assuming this rate in a human cell, there are 10,000 different sites in the body at which DNA is damaged every 24 hours. DNA has a number of quality control mechanisms. DNA polymerase, the enzyme that catalyzes DNA replication, has a proofreading function that immediately corrects 99% of these errors during replication. Those errors that pass through are corrected by a mismatch repair system. When a mismatch base, such as Ag, is detected, the incorrect strand is cut and the mismatch is removed. The gap is then filled in with the correct base, and the DNA is resealed.
Why do some animals only have a few offspring?
Females have a finite amount of energy that they can allocate to the growth and maintenance of offspring. Therefore, two basic strategies have evolved for successful reproduction. 1. Produce many small offspring, only some of whom may survive. Or 2. Produce just a few larger offspring, each with a higher likelihood of survival.
How do some cells, such as those found in nails and horns, change shape and become hard?
All of the surface cells of the body, except those in eyes, contain a molecule called keratin. A fibrous protein that is particularly well suited to withstand abrasions. Certain cells, like those of the nails and hair, have increased amounts of keratin that provide extra strength, helping them maintain their shape. Whether it is in the claws on the toes of your cat, the horns on the head of your favorite cow, or the 5 million hairs that cover your body, keratin strengthens and protects organisms from everyday wear and tear.
What is the relationship between blue jays, monarch butterflies, and milkweed?
As part of their life cycle, female monarchs lay their eggs exclusively on milkweed plants. After a few days the eggs hatch and a yellow, black, and white striped caterpillar emerges from each egg. These caterpillars are totally dependent on milkweed plants. Although the plants contain toxic substances, cardinalides, that are poisonous to other animals, the toxin is harmless to the monarch. Blue jays, who spend much of their day searching for food, will often eat insects such as adult monarch butterflies to supplement their otherwise vegetarian diet. However, if the food tastes bad, the blue jays will vomit up the food and will then learn individually to avoid the food in the future. Thus, wild monarch butterflies with high levels of cardinalide concentrations are less susceptible to natural predation by birds. This is an example of operant conditioning in the wild.
Who invented the electron microscope?
The theoretical and practical limits to the use of the optical microscope were set by the wavelength of light. When the oscilloscope was developed, it was realized that cathode ray beams could be used to resolve much finer detail because their wavelength was so much shorter than that of light. In 1928 Ernst Ruska, 1906 to 1988, and Max Knoll, 1897 to 1969, using magnetic fields to focus electrons in a cathode ray beam, produced a crude instrument that gave a magnification of 17, and by 1932 they had developed an electron microscope having a magnification of 400. By 1937 James Hillier, 1915, had advanced this magnification to 7,000. The 1939 instrument Vladimir Zworykin, 1889 to 1982, developed gave 50 times more detail than any optical microscope ever could, with a magnification up to 2 million. The electron microscope revolutionized biological research. For the first time scientists could see the molecules of cell structures, proteins, and viruses.
Can DNA be extracted from a mummy?
Yes, DNA can be, and has been, extracted from a mummy. However, the problem with extraction of ancient DNA lies within contamination from modern DNA. In order to minimize contamination, researchers usually try to get DNA from inside teeth or bone. Ancient DNA is being used to study the genealogy of the pharaohs of Egypt.
What is spectroscopy?
Spectroscopy includes a range of techniques to study the composition, structure, and bonding of elements and compounds. The different methods of spectroscopy use different wavelengths of the electromagnetic spectrum to study atoms, molecules, ions, and the bonding between them.
What does chest beating in gorillas signify?
Chest beating in gorillas is part of an aggressive behavior display. It is usually presented by a silverback, male, against unrelated silverbacks. A chest beating display, accompanied by hoots and barks, may also be used to impress females.
Examples of products and organisms made in bioreactors. What is a chimera?
The chimera from Greek mythology is a fire-breathing monster with a lion's head, goat's body, and a serpent's tail. The chimera of biotechnology is an animal formed from two different species or strains, that is, a mixture of cells from two very early embryos. Most chimeras used in research are made from different mouse strains. Chimeras cannot reproduce.
What animal makes the longest migration?
The Arctic tern, Sterna paradisea, flies a round trip from the Arctic of North America and Eurasia to the Antarctic. A round trip that can total as much as 20,000 miles, 32,000 kilometers.
What is the most common gene deficiency?
Glucose-6-phosphate dehydrogenase, G6PD, is the most common human enzyme deficiency in the world. It is estimated that about 400 million people worldwide are affected by this disorder. The enzyme G6PD is carried on the X allele and is therefore referred to as an X-linked trait. It is more likely to affect males than females. G6PD is a critical enzyme that catalyzes the oxidation/reduction reactions resulting in the production of NADF. NADF is a required cofactor in many cellular biosynthetic pathways. The stability of red blood cells is affected by G6PD deficiency. Since red blood cells are especially sensitive to oxidative stress. Clinical problems experienced by people with G6PD deficiency include neonatal jaundice and hemolytic anemia.
Is there a link between antibiotic use in animal feed and the increase in bacterial infections becoming resistant to antibiotics in humans?
Scientists have discovered a link between agricultural use of antibiotics, particularly in animal feed, and the increase of foodborne infections in humans who consume products derived from these animals. Resistant bacteria present in animals can survive the slaughtering and meat packaging process. Undercooked meat will harbor these bacteria and when eaten can cause illness in humans. To further complicate the situation, the antibiotics used to treat humans made ill by infections may be similar to those used routinely in animals, rendering them less effective.
Why do cells die?
Cells die for a variety of reasons, many of which are not deliberate. For example, cells can starve to death, asphyxiate, or die from trauma. Cells that sustain some sort of damage, such as DNA alteration or viral infection, frequently undergo programmed cell death. This process eliminates cells with a potentially lethal mutation or limits the spread of the virus. Programmed cell death can also be a normal part of embryonic development. Frogs undergo cell death that results in the elimination of tissues, allowing a tadpole to morph into an adult frog.
What is the lion hypothesis?
The Lyon hypothesis refers directly to a bar body. It was proposed by Mary Lyon, 1925, in 1961 that a bar body is actually an inactivated X chromosome. According to this hypothesis, female mammals sequester one X chromosome in each of their cells during the early stages of development. This folded chromosome becomes the dark body of Barr's observation. This means that both males and females rely on the information from only a single X chromosome. Therefore, it is only one X chromosome that provides genetic information in both males and females.
How does an antibiotic affect protein synthesis?
Antibiotics can slow or shut down protein synthesis in bacteria. There are about 160 known antibiotics, and each acts differently and selectively. Depending on the type of bacterial infection. As an example, streptomycin inhibits initiation of protein synthesis by interfering with final formation of the two subunits of a bacterial ribosome.
What is fermentation?
Scientists have theorized that fermentation was the process through which energy was first harvested from organic compounds. Fermentation evolved before Earth's atmosphere contained free oxygen. Aerobic respiration differs from fermentation in that the products of glycolysis enter the Krebs cycle rather than being used to form lactic acid or alcohol. Fermentation must have predated the appearance of oxygen in the atmosphere, which occurred over 2.5 billion years ago. Fermentation is thus an ancient process and occurs normally in microorganisms that live in the absence of oxygen.
Why are cells so small?
Cells come in a variety of sizes and shapes. Bacteria are among the smallest, 0.2 to 0.3 µm in diameter, while cells of plants and animals are generally larger, 10 to 50 µm in diameter. Cellular size is determined by the surface/volume ratio required to maintain access to the substances and enzymes that cells need to complete their functions. For further understanding of the surface/volume ratio, imagine that a cell is the shape of a cube. The volume of a cube increases as does the length or diameter, but the surface area only increases as the cube does. Once the upper limits of surface area/volume ratio are reached, the cell generates more cytoplasmic volume than can be met by the small increases in membrane surface area.
Can animals suffer from psychological disorders?
Although animals are used as models for research into disorders like anxiety, depression, and even schizophrenia, these conditions have been induced in them either through surgery or behavioral treatments. Scientists are currently developing strains of mice and rats that are genetically predisposed toward these diseases in hopes of gaining a better understanding of symptoms and more effective treatments. As for animals in the wild, the answer lies in the still unresolved issues of whether animals experience emotions and whether they have consciousness or the awareness of self.
What are the general characteristics of biomes?
A biome is one of the world's prominent ecosystems, characterized by both vegetation and organisms particularly adapted to that environment.
How are centigrade temperatures converted into Fahrenheit temperatures?
The formula for converting centigrade temperatures into Fahrenheit is degree F equals degree C x 9/5 plus 32. The formula for converting Fahrenheit temperatures into centigrade is degree C, degree F 32, x 5/9.
Did Darwin and Mendel know each other?
Although both men lived during the same time period in the 19th century, they did not know each other. Charles Darwin's, 1809–1882, publication on the origin of 387 species, 1859. Popularized his theory of natural selection but raised many questions as to how organisms could display modified or new traits. In 1865 Mendel published his landmark paper Experiments in Plant Hybridization. Researchers have been unable to document whether either man used the other's work in the development of their respective theories.
What is the optimum thickness for specimens to be examined under a microscope?
Biological and medical techniques require specimens of 1 to 50 micrometers, with the usual thickness being 4 to 5 micrometers for examination under a light microscope. Since an electron microscope has greater resolution, it requires thinner sections of 20 to 100 for biological specimens. Smear preparation is another standard technique used to prepare slides.
Where does the carbon dioxide that we exhale come from?
The carbon dioxide we exhale is a result of the breakdown of glucose during the energy harvesting phase of aerobic cell respiration. During this process, all of the carbon atoms, from the C6H12O6, are released as carbon dioxide molecules. Of the 6 carbon dioxide molecules generated, 4 are released via the Krebs cycle.
What is the difference between a virus and a viroid?
Viroids are small fragments of nucleic acid, RNA, without a protein coat. They are usually associated with plant diseases and are several thousand times smaller than a virus.
Why are some animals so brightly colored while others are so dull?
There are two general functions of bright coloration. The individual with such coloration is trying to advertise either to members of its own species, its conspecifics, or to those of other species. Within species, communication revolves around reproductive behavior. Male red-winged blackbirds, for example, use their brightly colored shoulder patches to advertise their territory ownership to potential mates and rivals. An experiment with zebra finches found that females were more likely to choose males as mates if the identification bands on their legs were red rather than some other color. Between species, communication is usually threatening. Warning, or aposematic, coloration is a method used by animals with stings or poison to circumvent attacks. For example, stinging insects like wasps and bees use similar color patterns of yellow and black to advertise their arsenal. Poison dart frogs, family Dendrobatidae, also use bright coloration in this way. Dull or camouflage coloration, as in a flounder, is an alternative strategy. By hiding, these species hope to avoid predation.
What is a daddy longlegs?
The name applies to two different kinds of invertebrates. The first is a harmless, non-biting, long-legged arachnid. Also called a harvestman, it is often mistaken for a spider. But it lacks the segmented body shape that a spider has. Although it has the same number of legs, 8. As a spider, the harvestman's legs are far longer and thinner. These very long legs enable it to raise its body high enough to avoid ants or other small enemies. Harvestmen are largely carnivorous, feeding on a variety of small invertebrates such as insects, spiders, and mites. They never spin webs as spiders do. They also eat some juicy plants and in captivity can be fed almost anything edible, from bread and milk to meat. Harvestmen also need to drink frequently. The term daddy longlegs also is used for a cranefly, a thin-bodied insect with long thin legs that has a snout-like proboscis with which it sucks water and nectar.
What is a bar body?
In 1949 Dr. Murray Barr, 1908 to 1995, noticed a dark body in the neurons of female cats. It was later identified as a structure found only in the nucleus of females. It was named a bar body in honor of its discoverer.
What is the most specialized cell in mammals?
Depending on the criteria you choose. Several types of cells could be nominated as the most specialized cell in the mammalian body. The two top candidates for this honor would probably be, 1. The cells that produce gametes, sperm and eggs, 2. The red blood cells that carry oxygen and carbon dioxide in the blood. Red blood cells are perhaps the most highly specialized. They only live for roughly 120 days. But during that time they may travel more than 500 miles 800 kilometers through various organs and blood vessels. Red blood cells lack a nucleus, so they are unable to reproduce. New cells are formed in bone marrow.
Why does the size of the nucleolus vary in cells?
Cells with a high rate of protein synthesis have a large number of ribosomes. In these active cells, nucleoli tend to be large and can account for 20 to 25% of the nuclear volume.
How do plant cells store energy?
Plant cells store energy in complex carbohydrates such as starch, disaccharides, and lipids. These energy sources may be used to fuel cellular metabolism or to provide energy for the germination of seeds.
Why are big and fierce animals rare?
Because the transfer of energy from one individual to another is inefficient. Only about 10% of the energy used to build a tasty worm actually is available to the hungry robin who eats it. As we move through the levels of the food web, each predator or predator grouping tends to become bigger and more aggressive, fiercer, than the last. However, the amount of energy available to each level continues to decline, so there is very little room at the top of the web for a predator large enough and fierce enough to consume all the others. In fact, it is estimated that only 1/1000 th of the energy brought into the system by photosynthesis actually makes it to the hawk or owl at the top of such a system. Less energy available means that fewer individuals can be supported. So big, fierce animals tend to be rare in their ecosystems.
What are buds on stems?
Buds may be terminal or axillary on a stem. The terminal bud is at the apex of the stem where the plant growth is concentrated. It contains developing leaves and a compact series of nodes and internodes. The axillary buds are found in the angles formed by a leaf and the stem. The axillary buds are usually dormant.
What is the difference between regular acidophilus milk and sweet acidophilus milk?
Both acidophilus milk and sweet acidophilus milk are inoculated with the bacterium Lactobacillus acidophilus. Many health practitioners believe that this bacterium, a normal member of the human intestinal flora, aids in digestion. Regular acidophilus milk is produced by adding the Bacterium to vats of skim milk as part of the fermentation process. It has a characteristic sour taste. When the bacterium is added to pasteurized milk and packaged without fermentation, sweet acidophilus milk, which lacks the characteristic sour taste, is produced.
What is a reading frame?
The reading frame is the 3-nucleotide section of the mRNA molecule that is read by tRNA during translation. Normally, the reading frame matches the codon, but mutations that cause the gain or loss of nucleotides can result in the offsetting of the reading frame, leading to the production of a very different protein. For example, a sentence comprised of 3-letter words changes its meaning with the deletion of one letter. The big fat cat ate one red rat. Delete the first letter E and the sentence becomes THBIGFATC at a tetrahedrat, which effectively is nonsense.
Which type of nitrogenous waste do various species of animals excrete?
Since it is highly toxic, excretion of pure ammonia is possible only for aquatic animals. Because ammonia is very soluble in water, urea and uric acid are excreted by terrestrial animals. Urea is approximately 100,000 times less toxic than ammonia, so it may be stored in the body and eliminated with relatively little water loss. Uric acid requires very little water for disposal and is often excreted as a paste or dry powder. An example is guano, the solid white droppings of seabirds and bats.
How is it possible to measure the pH of a solution?
An easy way to measure the pH of a solution is with pH paper. This paper is treated with a chemical indicator that changes colors depending on the concentration of H+, hydrogen ions, in the solution.
How dangerous is Clostridium botulinum?
The bacterium Clostridium botulinum can grow in food products and produce a toxin called botulinum. The most toxic substance known. Microbiologists estimate that 1 gram of this toxin can kill 14 million adults. This bacterium can withstand boiling water, 212 degrees Fahrenheit or 100 degrees Celsius, but is killed in 5 minutes at 248 degrees Fahrenheit 120 degrees Celsius. This tolerance makes Clostridium botulinum a serious concern for people who can vegetables at home. If home canning is not done properly, this bacterium will grow in the anaerobic conditions of the sealed container and create extremely poisonous food. The endospores of Clostridium botulinum can germinate in poorly prepared canned goods. So individuals should never eat food from a can that appears swollen, as it is a sign that the can has become filled with gas released during germination. Consuming food from a can containing endospores that have undergone germination can lead to nerve paralysis, severe vomiting, and even death.
What are the main organs of the digestive system and their function?
The digestive system includes the mouth, alimentary canal, or gastrovascular cavity, esophagus, stomach, small intestine, large intestine, and anus. The mouth is the opening through which food is ingested. In animals with a second opening for elimination, the digestive system contains an alimentary canal, a tube allowing for the passage of food from the mouth to the anus. In contrast, animals with only one opening have a gastrovascular cavity that serves as the site of digestive activities. The esophagus is another channel through which food passes on the way to the stomach. The stomach, or crop in certain species, such as birds, stores food and is the primary site of chemical digestion. Following digestion in the stomach where food is broken down with acid and enzymes, the food passes into the small intestine. Nutrients are absorbed via the intestine. Much shorter than the small intestine, though greater in diameter, is the large intestine, also called the colon. Here, the solid material remaining after digestion is compacted and then eliminated via the anus.
How do I know my enzymes are working?
Obviously, it is difficult to track the activities of the 20,000 enzymes that are required by your body. However, in the case of the enzyme amylase, you can easily check to see if it is working. Amylase is an enzyme in your saliva that starts the breakdown of complex carbohydrates into simple sugars, glucose and maltose. A plain cracker held in the mouth long enough will begin this breakdown, and you will actually begin to taste the sweetness of the enzyme products. Also, lysozyme is an enzyme present in your respiratory tract secretions and tears. It prevents invasion by bacteria, which explains why the warm, moist, open environment of our eyes manages to remain relatively infection-free during a normal day.
What roles do fungi play in recycling?
Fungi play a key role in the recycling of many elements. As the primary decomposers in the biosphere, they break down organic matter, including dead plants and other vegetation. As fungi actively decompose materials, carbon, nitrogen, and the mineral components present in organic compounds are released, these elements can all be recycled. During decomposition, carbon dioxide is released into the atmosphere and minerals are returned to the soil. It is estimated that, on average, the top 8 in 20 centimeters of fertile soil contain nearly 5 metric tons of fungi and bacteria per 2.47 acres, 1 hectare. Without fungi acting as decomposers, dead organic matter would overpower the world and life on Earth would eventually become impossible.
Why do birds flock?
The old adage about safety in numbers certainly holds true for flocking birds and other social groups. Flocks, herds, schools, and others provide a refuge for the young and an anti-predator advantage for all. The simultaneous movements of a large number of prey are apt to confuse predators and decrease the likelihood that they will capture any one individual.
Which bacteria were used to clean the beach after the Exxon Valdez oil spill?
B. Bacteria from the genus Pseudomonas are capable of degrading oil as their source of nutrition. This microorganism was used to clean up the spill that emerged from the Exxon Valdez. In order to improve the efficiency of this bacteria, nitrogen and phosphorus were introduced to beaches polluted by the spill. The number of oil-degrading bacteria on the beaches increased sufficiently enough to remove much of the oil from the beaches.
Where do scleriids occur in plants?
Scleriids occur in the roots, stems, and seed coats of plants. Groups of scleriids in the soft flesh give pears their characteristic gritty texture. Scleriids also give nutshells and seed coats their hardness.
How is the Cambrian explosion related to Evo Devo?
Mutations in the suite of genes that control developmental processes, known as the Hox genes, would have caused the radical changes in the body structure of animals that occurred during the Cambrian explosion. These genes have been identified in every major animal group, both vertebrates and invertebrates. So it seems likely that they date back to the Precambrian period. Thus, the mechanisms that control the development of the animal embryo, devo, also probably played a role in the evolution of animals as a group, thus the term evo-devo.
How do sperm work?
Cells called spermatogonium become sperm through the process of meiosis. A mature sperm has only half the DNA required for a functional cell, so it can't survive on its own. However, they do have flagella and mitochondria that power their journey through the reproductive tract in search of an egg. Human sperm typically die within 48 hours if they have not fertilized an egg.
Why are sea urchins used to study embryonic development?
Sea urchins are a useful model system for studying many problems in early animal development. Historically, sea urchins were a key system in elucidating a variety of classic developmental problems, including the mechanisms of fertilization, egg activation, cleavage, gastrulation, and the regulation of differentiation in the early embryo. In addition, early studies of the molecular basis of early embryology were carried out in this system. Gametes can be obtained easily, sterility is not required. And the eggs and early embryos of many species are beautifully transparent. The early development of sea urchin embryos is also highly synchronous, that is, when a batch of eggs is fertilized, all of the resulting embryos typically develop on the same schedule, making possible biochemical and molecular studies of early embryos.
What is transduction?
Transduction is the process by which a vector, usually a bacteriophage, carries DNA from one bacterium to another bacterium. It can be used experimentally to map bacterial genes.
What is a pedigree?
A pedigree is a genetic history of a family, which shows the inheritance of traits through several generations. Information that can be obtained from a pedigree includes birth order, sex of children, twins, marriages, deaths, stillbirths, and pattern of inheritance of specific genetic traits.
How has biotechnology helped thalidomide become popular again?
The drug thalidomide was developed during the 1950s. It was found to be effective in combating the symptoms of morning sickness in pregnant women. unfortunately, if taken during the first trimester of pregnancy. Thalidomide causes serious limb deformities because it interferes with the normal growth process of the fetus. Recently, it was discovered that the drug also has the ability to interfere with the replication of HIV in human cells. Thalidomide also may prevent the weight loss commonly associated with AIDS. Thalidomide actually has two mirror image forms, chirality, only one of which causes birth defects. Researchers hope that by using biotechnology they will be able to separate the forms and return thalidomide to their arsenal of disease-fighting chemicals once again.
Why is the shape of an enzyme so important?
Shape is critical to the function of all molecules, but especially enzymes, which are three-dimensional. The active site of an enzyme is the area where substrate binds and the reaction takes place. How an enzyme reacts with its substrate is similar to how a ship docks. There are minor bonds that form between the enzyme and substrate until docking is complete. Anything affecting a protein's shape would have an effect on its ability to react with the substrate.
What disease is caused by the protist Entamoeba histolytica?
Entamoeba histolytica, a parasitic amoeba, causes amoebic dysentery, an intestinal disorder. Estimates indicate that up to 10 million individuals in the United States have parasitic amoebas. But only 2 million exhibit symptoms of the disease. In tropical areas, up to half the population may be infected.
What is the standard procedure for differential centrifugation?
Differential centrifugation is a technique commonly used by biochemists. Tissue, such as liver, is homogenized at 32 degrees Fahrenheit 0 degrees Celsius. In a sucrose solution that contains both a buffer to stabilize the pH and a salt. The homogenate is then placed in a centrifuge and spun. At a constant centrifugal force at a constant temperature. After a predetermined amount of time, a sediment forms at the bottom of the centrifuge tube and is covered by an overlying solution called the supernatant. The overlying solution is then placed in another centrifuge tube. The sediment after the first centrifugation is called the nuclear fractions and consists mainly of nuclei, which are the largest and densest organelles present in cells. The supernatant is centrifuged at a higher centrifugal force for a longer period of time. Another group of particles, usually the mitochondria, form the sediment. The process is repeated several times, each time at a higher centrifugal force for longer periods of time, until the sediment only contains enzymes and other substances not associated with any cell organelles.
What is a mutation?
A mutation is an alteration in the DNA sequence of a gene. Mutations are a source of variation to a population. But they can have detrimental effects in that they may cause diseases and disorders. One example of a disease caused by a mutation is sickle cell disease, in which there is a change in the amino acid sequence. Valine is substituted for glutamic acid. Of two of the four polypeptide chains that make up the oxygen-carrying protein known as hemoglobin.
Is there a relationship between the size of the root system and the size of the shoot system?
Growing plants maintain a balance between the size of the root system, the surface area available for the absorption of water and minerals, and the shoot system, the photosynthesizing surface. The total water and mineral absorbing surface area in young seedlings usually far exceeds the photosynthesizing surface area. As the plant ages, the root-to-shoot ratio decreases. Additionally, if the root system is damaged, reducing the water and mineral absorbing surface area. Shoot growth is reduced by lack of water, minerals, and root-produced hormones. Similarly, reducing the size of the shoot system limits root growth by decreasing the availability of carbohydrates and shoot-produced hormones to the roots.
What molecules are most important to living organisms?
Four molecules are referred to as bioorganic because they are essential to living organisms and contain carbon, nucleic acids, proteins, carbohydrates, and lipids. These molecules are all large, and they are formed by a specific type of smaller molecule, known as a monomer.
What is the most expensive spice in the world?
The world's most expensive spice is saffron. The spice was highly sought after by the ancient civilizations of Egypt, Assyria, Phoenicia, Persia, Crete, Greece, and Rome. The term saffron comes from the Arabic word za'faran, meaning yellow. The spice is obtained from the delicate stigmas of an autumn crocus. Crocus sativus, a species native to eastern Mediterranean countries and Asia Minor. Crocus sativus is propagated by corms. The blooming period for the crocus is approximately 2 weeks, after which the flowers must be picked while they are in full bloom and before any signs of wilting. Once picked, the 3-part stigmas are removed from the petals before the petals wilt. This is a time-consuming process that can only be done by hand as the stigmas are very fragile. Then the stigmas are roasted and sold either as whole threads, whole stigmas, or powder. In order to harvest 1 pound 0.45 kilograms of the spice, between 75,000 and 100,000 flowers must be picked. Approximately 4,000 stigmas yield 1 ounce, 28 gr, of the spice. In 1998 the retail price of saffron was approximately $240 per ounce. $8.50 per gram, confirming its place as the world's most costly spice.
What distinguishes the different types of microscopes?
Microscopes have played a central role in the development of cell biology, allowing scientists to observe cells and cell structures that are not visible to the human eye. The two basic types of microscopes are light microscopes and electron microscopes. The major differences between light and electron microscopes are the source of illumination and the construction of the lenses. Light microscopes utilize visible light as the source of illumination and a series of glass lenses. Electron microscopes utilize a beam of electrons emitted by a heated tungsten filament as the source of illumination. The lens system consists of a series of electromagnets. Recent advances using optical techniques have led to the development of specialized light microscopes, including fluorescence microscopy, phase contrast microscopy, and differential interference contrast microscopy. In fluorescence microscopy, a fluorescent dye is introduced to specific molecules. Both phase contrast microscopy and differential interference contrast microscopy utilize techniques that enhance and amplify slight changes in the phase of transmitted light as it passes through a structure that has a different refractive index than the surrounding medium.